Today I'll be discussing reducing gene expression in eukaryotic cells. I will be looking specifically at RNAi method versus the CRISPR method. So RNAi is ribonucleic acid interference. It involves siRNAs, which are small interfering RNAs, usually about 20 to 25 base pairs long. You have a target mRNA that you would like to degrade, and then you have this RISC complex, which is RNA-induced silencing complex. And so this is actually what will cleave the target mRNA. So basically you have a designed siRNA molecule that is designed to be complementary to your target mRNA strand, and this will attach to the RISC complex, and then it will actually go and find and hunt the target mRNA and will attach to it using complementary strand, and it will actually cleave it. And now this mRNA will not be able to be used to make proteins. So you've effectively reduced your gene expression. I'll now be discussing the CRISPR method. So CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. Again, due to time constraints, this will be a very simplified version of how this happens. But basically you have an sgRNA molecule, which is your single guide RNA molecule. I've illustrated that here. And so this will be designed to be complementary to your target sequence of DNA, which I've illustrated right here. And then you have a Cas9 endonuclease, which is just a CRISPR-associated endonuclease. And this is an enzyme that will actually cleave the DNA that you've specified. So this complex will find your target sequence using the complementary sgRNA that you've designed, and the complex will attach and then cleave the DNA. And this is where researchers could either insert a new sequence, modify an existing sequence, or remove an existing sequence in order to reduce or affect gene expression. When trying to determine which one of these gene editing techniques would be better, it really depends on the experiment that you're running and what you're attempting to do. In this case, I would choose RNAi, simply because, generally speaking, it's faster, it requires less cloning work in the lab, and the results can be measured fairly easily using a QRT-PCR assay, which would measure your RNA levels, or a Western blot assay, which would measure your protein levels. One potential drawback of the RNAi method is off-targeting. One molecule of siRNA may suppress many different mRNA transcripts, which basically means that you could be suppressing other gene expression that you aren't intending to suppress. But that was not addressed as a serious issue in the prompt, so I would go with RNAi in this situation.